Okay, welcome to week 3. In this next week, we're going to cover extensions and exceptions to Mendelian genetics. This is primarily going to be covering chapter 4. So before I begin, I just want to remind you that you should review chapter 3 from our genetics book as well as chapter 11 from the Biology 111 book. And I want you to just review basic Mendelian genetics, so make sure that you have it down pat, because now when we do some exceptions to the rule, I want you to have the basics down before you start introducing some newer ideas. Okay, so there are several different types of alleles or mutations that we can get. The first we'll call wild type. And so this is going to be the version of the allele that's going to be most frequently found in a particular population. We're typically going to call this normal, but just know that this normal is kind of arbitrary. The only reason we call it normal is because that's what we find the most of. The next type you might find is a loss of function, sometimes written LOF. This can result in reduced activity of the given gene product. We will call this a null allele if the function is completely lost. Sometimes we might get a mutation that's going to be a gain of function mutation. And in this case, you're going to be looking at things like proto-oncogenes. So these are going to be genes that if they get mutated, they may do things like regulate the cell cycle in weird ways, and this can lead to different types of tumors to form. We can also have neutral mutations. So depending on what happens, it might give you either no change to the phenotype or just no change to the overall fitness of the organism. Say in humans, there was a mutation that gave somebody purple freckles, right? That's not going to affect how well that they live their life. It's just going to be a mutation that happens. Geneticists are going to use a variety of symbols for alleles. And typically, when we talk about our dominant alleles, as we've done previously, they're going to indicate them as an italic uppercase letter, sometimes as a group of letters like WR. And recessive alleles are usually indicated by either a lowercase letter such as a D, an italic letter or group of letters with the plus superscript, so WR plus. When we're talking about genes, typically everything is going to be italicized, and that's where the italics are coming from. Sometimes, as you'll see with my stuff, they might be a little bit lazy, but when you're talking about something that's going to be a published work, it is, or it should be, italicized. There are some cases where no dominance is going to exist. There's just different versions or alternative alleles. In that case, what's going to happen is you're going to have italic either uppercase letters or superscripts. So in here, in this case, it's numbers, this case, it's letters, just to denote what the alternative alleles might be. In Drosophila, here's some examples of how we indicate alleles in Drosophila. So Drosophila melanogaster, a very commonly used model organism, particularly in genetics. One example could be body color. So ebony is a mutant phenotype, and we're going to indicate that with a lowercase italic e. Normal gray or wild type is going to be indicated by this E+. So if something was a gray homozygote, wild type, it'd be E+, E+. These should be italics, but that's okay, again, lazy. If it's a heterozygote, you can see the E+ slash E. It's still going to have the wild type phenotype, but in this case it's going to be a heterozygote because you're going to have the two different copies. This one here, just the E is going to be the ebony homozygote, and so this is going to be the mutant organism. Another way of writing it, sometimes they'll even do another shortcut. They'll just do plus plus if something's going to be wild type, the plus meaning the wild type. In this case, it's be homozygous. If it's a plus E, you could say that it was a heterozygote, but it's still going to be wild type phenotype. Lowercase e is going to be the ebony homozygote or the mutant. Okay, so let's talk about some more rules of genetics. There are cases where we have an incomplete or a semi-dominance of different alleles. And when we see this is when the heterozygote is going to have an intermediate phenotype between that of either homozygote. So one of the classic examples for this are going to be snapdragon flowers. So in the first parental generation, We can see here R1, R1, we're going to have red flowers. Here would be R2, R2, and these are going to be white flowers. If you were to cross these together using typical classical Mendelian genetics, you would expect 100% of the offspring to be red because that red's going to be dominant. However, in this case, it's just a little bit leaky. The heterozygote has this intermediate phenotype very easily seen as a pink color. So we're going to have R1 from the red, R2 from the white in the F1 generation. Now, looking again at these flowers, okay, so we've got this parental generation. In this case, they're using the lowercase r's and the uppercase r's. The F1 generation, all going to be heterozygotes. We know that it is not just the simple blending of traits because there's two different types of crosses that we can do. First, if you were to cross this F1 generation, cross a pink with a pink, the other way we can tell is if you cross this pink with a white. We're going to see very distinctive ratios of outcomes. And that is, if you cross the pink with the pink, about a quarter of them are going to be white, which is back to the grandparents' generation now. About 50% or 1 in 2 of them are going to be pink because they're going to be heterozygotes. And about 1 in 4 or 25% of them are going to be red because they're going to be homozygous dominant for both alleles. If we were to do the test cross here, the heterozygote times the homozygous recessive, what you'll see is about 50% of them are going to be pink and about 50% are going to be white. And so these data indicate that this is not just a simple blending of traits because we can get those original grandparent phenotypes back out from these crosses. There are examples of incomplete dominance in humans, and one of these would be the example of Tay-Sachs disease. So in this particular disease, these homozygous recessives, if you're homozygous recessive, you might die from this fatal lipid storage disorder. And there's an enzyme, this hexosaminase activity is going to be absent. Heterozygotes are going to appear normal, but they have actually half the amount of the wild type enzyme activity when you compare them to the homozygous normal non-carriers for this trait. In this case, this is sufficient for normal health. Luckily, that 50% activity is sufficient for normal health, and these individuals who are carriers are phenotypically normal. This demonstrates the threshold effect. What happens is if this normal phenotypic expression occurs whenever a certain level, maybe 50% or less, of the gene product is attained. So as long as you have at least about 50% or so gene activity, the individual is going to present as phenotypically normal. Now, if for some reason they have a copy where it's not working quite as well, maybe they're only having 25% activity, now these individuals might be affected. Another example that we might see is codominance. And what happens here is you have two alleles at a locus. They're producing different detectable gene products in the heterozygote, but there's no case of dominance or recessiveness, and you're not going to see a blended phenotype. So you're not going to see this incomplete dominance. So an example of this would be blood type in humans. So the A blood type, the B blood type, they're actually codominant with one another. And an individual might exhibit both of those. So you can have individuals who are AB blood type. So in this case, you're expressing both A and B. A is not dominant to B is not dominant to A. They are both being expressed. And one is not masking the expression of the other. So this segues nicely into our multiple alleles. And so although any individual has at most two different alleles, one from their mother and one from their father, most genes exist in a large number of allelic forms in the population as a whole. And so multiple alleles is only going to apply when you have 3 or more alleles or versions of the same gene in a population. So looking at the example of blood type, there are 3 different possible versions that individuals might get. One is A, one is B, and one is O. So A and B are going to be dominant to O. So if an individual has 2 A's, they're going to have blood type A. If they get an A and an O, A is going to be dominant to B. These 2 genotypes are going to produce indistinguishable group phenotypes, and they're both just going to be typed as type A. If an individual gets 2 B alleles or a B allele and an O allele, they're going to produce type B blood. If they get an A and a B, they're going to be type AB blood, and if they have to get 2 O alleles in order to get type O blood.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
So when we look at Punnett squares for individuals with different blood types, again, A and B are considered to be codominant or incompletely dominant to one another. If we were to cross an individual who has type AB blood with an individual who has type O blood, type A blood with an AO genotype, what we would expect to see out of this particular cross is about 25% or 1 in 4 of the offspring would be type AB. About 1 in 4 of them would be type B, and a 2 in 4, or 1/2 of them, would be type A phenotype. If we were to cross an individual who was type A blood, but genotypically AO, with an individual with a genotype of BO, the types of offspring we would expect to see from this cross, we would expect to see about 1/4 of them to be type AB, about 1/4 of them to be type B, about 1/4 to be type A, and about 1/4 of them to be type O. So there's an interesting case study, they call it the Bombay phenotype. There was a woman who was typed as blood type O, but she had one parent with type AB blood, which shouldn't be possible, and she was an obvious B allele donor to 2 of her children. So this was a very strange case, but what they found was that she happened to be have a different mutation, and this mutation caused her to make no substrate to make the A or B antigens. And so although she may have been type A or B, or maybe type AB, we don't know, but she didn't have the enzymes required, the substrate required to make those antigens. And so because she didn't make them, those antigens, they showed up as type O. And we can see the family tree here. Her typing as blood type O, it might sound like, oh, maybe, it's not quite the parent of the child that they thought, maybe there's something fooling around going on. But since the mother was type AB, there is no O possible for her to have inherited. And then the same with the children. Because she was the mother, she had children with a type AB blood, type B blood. That shouldn't have been possible. The only reason it was possible, and the only reason she showed up as type O, was because she was not making the enzyme to make these different alleles. And in fact, it's probably likely that she was either type AB or type B blood.